Hello, 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 everyone. Welcome back. I'm on again with Standing for Truth, and we're going to talk about stuff and things and stuff. Mostly biological things and stuff. Maybe fossil record things and stuff. We might get into those, I'm hoping. But we'll see. So last time we did our talk, which was a few weeks ago. Uh, I said a few things which were slightly erroneous. For instance, I either said or implied that the feathers on Archaeopteryx were, hey, Beej and James and Rayrard and Joel and everybody else, that the feathers on Archaeopteryx were modern, or that modern feathers preceded Archaeopteryx. Neither of those are in fact the case. Modern feathers didn't come along until the Cretaceous, and Archaeopteryx's feathers were primitive with respect to modern birds. So yeah, that was something I wanted to point out there. If there's anything else I mistakenly said last time, then be sure to point it out to me, and I will correct it post haste because I hate to be wrong. But when am I ever wrong? Ha ha ha. Anyway, so if you were here last time, then you know Standing's position. But in case not, I'm going to turn over to him and let him explain. So go right ahead.
Awesome. Thanks for having me on your show again, Jackson. I think we had a good, interesting, profitable discussion last time. I look forward to talking about, I'm sure we'll rehash some of the things, also get into some other, important evidence, whether it's Chromosome 2 or, whatever it is. I'm sure it's going to be interesting. Yeah, I mean, my position's pretty simple. I hold to a biblical model. We believe that obviously God created, species not in, a static position. Obviously, neither their genomes. We believe that God would have created, the original kinds, including Adam and Eve, with, tens of millions of nuclear DNA differences. So with all that, heterozygotic DNA variety, going from, heterozygosity to homozygosity would allow for that, extensive variation that we see around us. And that's why, according to our model, the millions of species, in the 6,000 years, it was known as, a major problem. But I mean, with those tens of millions of DNA differences, and all those heterozygotic gene and DNA sites, genetic calculations would suggest, based on these differences from the start, and with these massive amounts of DNA variety, say encoded into, each kind at the start of their existence, really the potential for speciation's mind-boggling, right? I'm sure we'll get into a discussion on speciation, but combined with the fact that species can recover enormous population sizes in even very short amounts of time, in addition to the fact that, the vast majority of these DNA sequences within a creature appear to be functional. And I know we touched on that last time, and I'm sure we will this time, with the junk DNA and the AREs and the ERVs. These results would demonstrate that millions of species in a few thousand years is not only plausible, but also probable. And combined with, the fact that our model would predict limits in the genome and in the variation we see, we would expect, the process of devolution, known as genetic entropy, to be taking place, that accumulation of near neutral mutations. I use the word near because, according to our model, we do predict that, the majority of the DNA sequences are functional, whether it's, I understand that these non-coding, non-protein coding regions, are known now to have expressive roles, placental and embryological roles. So that means that, these mutations, whether positive, neutral, or harmful, they're going to have deleterious effects. So I think it all comes down to a net loss versus a net gain. And a lot of these mutations, they're obviously environmentally dependent. So, I mean, it might be beneficial, as in a beneficial outcome in one environment, but at the end of the day, it's deleterious in another. So, I mean, that wouldn't, satisfy the requirement, say, for a net loss versus a net gain of information. And I think empirically that's what we see. you know, if we see this degeneration and entropy process going on, you know, we could trace that back to a point of least entropy, which would be a point of creation. And to me, that's what makes sense. And I think, you know, going into these things will be, you know, a fun discussion. I'm good.
Okay, so a lot of things. First, really first and foremost thing that I really want to hit is because we talked about the notion of information last time, and I'm still not satisfied with what you mean, and what you said just now raised the hairs on my neck quite a bit. Because you accept that natural selection works, right?
Correct.
And organisms
I would say natural selection is more of a fine-tuning process, and that's why, like, when it comes to genetic entropy, I believe that selection is going to amplify, you know, the most beneficial of mutations, and obviously it's going to, you know, get rid of the most harmful. But it's the near neutral mutations that I believe are the major problem, and the problem with these deleterious effects that we're seeing. So that's why I think it all comes down to genetics. I think the debate
I absolutely agree.
is a genetic debate at this point, right?
Oh, absolutely, and that's why I wanted to ask you. So you do accept the natural selection occurs? I mean,
I do. I do.
And so, I mean, you know, the major creationist organizations, ICRA, IG, CMI, they all accept that natural selection occurs. So you say that these things that even if they gain something in one environment, then that wouldn't help them in some other environment. Is that an accurate reflection of what you said?
I would say that, you know, mutations are environmentally dependent. So yeah, in one area, like sickle cell anemia, for example, you know, it confers a resistance to the malaria parasite, but at the end of the day, you know, it's producing deformed hemoglobin molecules. So it's going to be beneficial, you know, to somebody who, you know, might get malaria, but at the end of the day, it's still detrimental and it's still deleterious. Actually, are you familiar with the HIV example? Because I noticed, like, would you use that as an example for, say, novel information?
HIV in what way?
It's an example where I noticed in the comments section, I think it was from a guy named Frustrated Atheist. He used that as, you know, the definitive example for new novel information. It's a mutation that prevents HIV from entering the white blood cells.
Oh, but it's another heterozygotic advantage, isn't it? Where it confers one benefit, but if you have both versions of the allele, then it is harmful, isn't that right?
Exactly. So it actually.
Well, that's why I avoid those examples. That's why I avoided those examples last time we talked with, for instance, the trypsinogen protein that was duplicated to become the antifreeze protein, or the example of the duplication of Geisha and mother of Geisha in Ragavalia, which created the novel propeller fans, which allowed them to invade a new environment.
No, that's why I do, and use the melanoprotein example as well, that I think helped with the cholesterol. Right. I think with the antifreeze example, because even looking at the gene that was enhanced, you know, it was really just enhancing, and correct me if I'm wrong, and give me your thoughts on this. It seems like it's just enhancing a pre-existing antifreeze function.
It wasn't. That's the wrong kind of antifreeze protein.
Is that the SAS gene?
Not the one that I'm referring to. The one you're referring to is in zooarcid fish. The zooarcids, in the case of the zooarcids, yes, it was just an amplification of the SAS, the sialic acid, whatever it was, gene, and which already had a minor antifreeze role, and it basically, and in the zooarcids, it basically just duplicated, and so it just became more and more powerful at its job. In the case of the notothenidoids, you have an example.
I just want to ask you, would you use that example, though, as an example, say, for molecules from Anpons come to people?
I still want to get back to the major point that I was trying to get at originally. We can talk about the individual mutations, but I want to get at the central point.
Yeah, because it looked like you wanted to talk about, like, information and definition of it, right?
We can. I want to clear up what I think is a misconception, or maybe I misheard you. I could have misheard you.
No problem. Go ahead.
The mutations are environmentally dependent. I absolutely agree with that. All mutations everything is environmentally dependent. Nothing could ever not be environmentally dependent, right? Because an African elephant doesn't live in Antarctica or the tropical rainforest. It lives on the African savanna, right? So
Correct.
every mutation that affects it is only going to matter to its environment. It's not going to matter to polar bears or to, jaguars in South America.
Well,
And and Kent Hovind said something similar when I talked to him, and it's something I didn't, or something I at least I think I should have hammered on back in the day, but I didn't. I don't remember a whole lot from that conversation, but that's one of the few things I remember, is that mutations are always environmentally dependent. They have to be. They can't not be, in a sense. And so it doesn't matter what's happening over in some other part of the world. As long as it benefits you in your environment, that's beneficial to you.
Well, I think it's not exactly, that it's, another part of the world. It's just that if this, like, say, new trait or this amplification that's occurring, if it's, say, a reduction in specificity, say with, nylon digestion by bacteria, or if it's, kind of like the example with the, because even the melanoprotein is a reduction in specificity as well. It's still deleterious.
Sorry, it's a reduction in specificity? What does that mean?
So it's reducing its, specific, say, function. So it's reducing specificity.
Its function. In the case of the apolipoprotein A1 melano, what it did was it replaced It replaced one of the amino acids with a cysteine. I can't remember which one it was.
No, yeah, it was a cysteine. Because I remember, I think it was a cysteine in a protein that normally assembles the HD, like the high density lipoproteins. They're involved in bad cholesterol, right?
It was what I mean is it was a point mutation that caused it to, in essence, enhance the function of this, because it already had a function in this, in helping us metabolize cholesterol, bad cholesterol.
And I totally agree, but I'm just saying that this, this amino acid that's been replaced, originally its job was actually to remove the bad cholesterol from the arteries. So now
it's doing that better now.
No, because the mutant form of the protein is now less effective of what it was supposed to do.
No,
now it's not acting on the antioxidant, which presents the problem is atherosclerosis, right? Hardening of the arteries. So you're getting this new trait, but it's due to a loss of specificity, okay? Because its original function is now reduced, because it's important to get rid of that bad cholesterol. And at the end of the day, that example is not really, to me, a strong example, because just with a healthy diet, exercise, you can reduce your risk of, the heart disease that apparently this mutation is helping against, right?
Actually, if you look at the technical literature on it, what it says is that it increases their ability to metabolize the bad cholesterol. It increases, I can't remember what it was specifically, but their cholesterol is not being raised, the bad cholesterol. So they're able to intake more levels of bad cholesterol and not suffer things and they're less likely to suffer things like you said, heart disease, things like that. And so this is an example of, and so sure, you can say it's an amplification of this thing that already had a function in it, but it's doing it better now. And as we kind of covered a little bit last time we talked, evolution is all about co-option of structures.
On this one, I just want to ask you, do you believe that this is also a heterozygotic change? Because to me, it seems like, well, for example, much like with sickle cell anemia, right? Like it's a variation of the pre-existing information. Because the fact, like what you were saying just now, Jackson, the fact that the person, say, with this mutation, they're now unable to produce the normal levels of HDLs.
Can you explain me what a heterozygote is?
So are you talking about like a heterozygotic gene because if you have that, say,
yes, if you are heterozygotic for a gene, what does that mean?
You have a lot of potential for variation, because we say
that's not what that means.
What does it mean?
So So you know what alleles are. You have alleles of variation of a gene, right?
Of
You know what a
course,
punnet?
yeah. Like I could have an allele for various different eye colors or various different hair length. Like there's multiple variations per gene at the loci, right?
Okay.
At the position.
So okay, right. So that's what heterozygotic is. It doesn't mean you necessarily have the potential for variation. It means you are carrying two alleles, two different alleles. You're not carrying the same allele, which would make you homozygotic. It doesn't have really anything to do with
Well, like if there's
carrying the necessary variation, because
well, like
ultimately, when you have a child, you're only going to pass on. You're going to pass on the the chromosome. You're going to pass on only one of your eggs and one or sperm also, and so you're going to have the you've got it's diploid. Your chromosomes are diploid, so you have the two different versions of the same gene, which are your alleles, and you're only going to get
I understand what you're saying, but I'm saying like if there's inbreeding going on, you're going from a shift from heterozygosity to homozygosity because now there's less variation possible in that population.
I mean, it's a lot less about
potential, right? It's
not
if you're
about
more
potential for
heterozygotic,
variation.
you do have more potential because we would say Adam and Eve had more heterozygotic DNA, which allowed for the variation.
When they had a child, how many of their alleles for any specific gene did they pass on to their child?
Well, how many different, like for example, at the loci, how many different locations are there on that, say, specific gene?
So for eye color, for instance, how many alleles for eye color did they pass on to their child?
But isn't it based on a certain location on that gene? Like, is it two alleles?
Yes, two alleles.
Yeah.
They would have passed on only two, meaning that however many God created them with, they passed on just two. So when you get to the flood bottleneck, it doesn't matter if you have, it doesn't matter how many alleles, God put in each elephant or each pine tree or whatever. Ultimately, they're only going to pass two on to their offspring.
But what I'm saying is those nuclear DNA differences that we see among species, we say most of that is actually based on the creative handiwork of God at creation. He put those tens of millions of DNA differences into each created kind. That's why I say, we have a lot of compressed, overlapping information in our code. In hidden compressed form, right? So over time, based on mutations and natural selection, we're seeing a lot of these traits. A lot of this variation is all based on that pre-existing information that God placed into each animal.
So, for instance, there was an elephant kind that got off the ark, right? There were only two elephants of the elephant kind, whatever they may have looked like, that got off the ark. Right?
I like to look at the kinds as more of like a gene pool. Okay, so you're going to have a strong gene pool.
Got off the ark.
I would say like elephants, mammoths, mastodons. They all descend from a single common ancestor that would have been
but that is the kind, right? That's the holobaramon, whatever the whole group is.
Exactly. Yeah. Whatever the whole. Okay, but the thing is, I can't exactly identify what that original.
Well, no, you don't have to. I'm not asking you to. I'm just
okay. Yeah.
No, I'm not asking. I'm just making a point about how how many two elephants, whatever they may have looked like, they gave birth ultimately to the rest of what we consider the elephants. Eventually, the mammoths, mastodons, etc. So they did that.
They all share a common ancestor with that original kind, of course.
So the elephant kind, the two archetypal elephants who got off the ark, how many alleles would they have passed on to their offspring?
The thing is, are you saying there's two alleles per gene, right?
Because we're diploid, yes. Right? There are two. Yes, per loci, yes. So you have two. They would have passed two, meaning that if they're just passing two, even if they have huge numbers of alleles within them, which I don't know how that would be possible, but let's just assume it is. They're passing two on to each of their children, meaning that somehow to go from the variation from there with just two, 4,000 years ago, to the variation in alleles today would have required magnitudes faster mutation and radiation than anything we've ever seen or indicated by.
Well, the thing is, like I said at my opening statement, based on those tens of millions of DNA differences, that would not be a problem at all, because we're seeing a sorting out of this information. Because at the end of the day, you know, let's say with new species, okay, you know, losing the ability to interbreed, say each new species that we see now, they're going to have less genetic variability, which means less ability to meet changes in its existing environment. It's all based on a sorting out and a loss of genetic information, because according to genetic entropy, we've degenerated, we've accumulated these near neutral mutations. Take that back in time. You can't look at species today, like an elephant population, and see that sorting out process. You got to take that back into time and see the tremendous variability and potential for that variation.
Shouldn't we be able to, based on, I mean, that's how kinds are built in a sense.
Well, we can see that you know, the majority of this non-protein coding nuclear DNA sequences are now showing function. So that's
they're not, and we talked about this last time. I explained that they're not showing function
because you're trying to say that embryological, placental functions, gene expression type functions, regulatory functions, those are functions, which means if there's a mutation in that gene, it's going to be deleterious. I mean, how do you just ignore that?
Well, first of all, I'm not ignoring anything. What I still want to get back to is the point about how what you're asking for when you have the kinds who are getting off the ark, whatever they may be, elephant, pine tree, whatever, the parents are passing on 2 alleles per loci, which means that somehow to get the variation we have just 4,000 years later, magnitudes faster radiation is going on in these populations than has ever been observed or can be inferred from the genetic data. So how do you explain that away?
I already explained that these tens of millions of DNA differences were placed into each created kind. It's not a problem because it's been a sorting out and a loss of this genetic information. The thing is, Jackson, you need to realize that I think this is a valid explanation, but you're trying to say that all the animals we see today came from 2 animals ultimately. So, I mean,
that's which what your model is proclaiming. That's what your model proclaims, because they got off the ark and it was just 2 elephants who got off the ark.
Because you're trying to say that we've all, you know, descended from a single or maybe some multiple common ancestors. But I'm trying to say that, you know, there were created kinds that were able to bring forth and descend all this variety.
So I'm confused. So are you saying that there weren't originally these common ancestors who got off the ark and radiated into a number of different groups?
Well, of course, yeah, through genetic drift, through, you know, mutations, natural selection, migration.
Okay, so where did the genetic variability come from? Because the parents are not passing this on. They severely limited, they bottlenecked. This is an insane bottleneck, again, one which isn't inferred in any species, and passed on 2 alleles per offspring, and somehow we're getting the huge genetic variety in these populations today over just 4,000 years. That's not explainable by invoking genetic entropy.
No, because I'm saying, well, just wait. I'm saying that mutations, right? So changes in the DNA that occurred after man's sin, okay? So after the fall, but also after this bottleneck of the flood, you know, it corrupted God's creation, but it didn't produce new genes. Rather, the mutations only produced the alleles that you're talking about, which is just variations in the pre-existing genes. So with all that potential for variation and a point of least entropy, I mean, I don't know why you think it's such an issue that, you know, over those thousands of years, whether it's 6,000 or 4,500 since the flood,
okay,
that sorting out of pre-existing information is no problem.
Okay, so you've got elephants. Let's stick with elephants. And you've got the elephant parents who passed on, and they may have millions. Somehow they've got millions of alleles magically, but they've got to pass these on naturally following the global flood. Meaning, unless God decided to inject all the following elephants with millions of alleles, again, somehow they've got to. I don't know how that would work. I mean, none of it makes any sense because you've got.
Well, how large or how wide is the gene pool of an elephant?
So what happens in genetic bottlenecks is that genetic diversity is highly reduced. When you look at cheetahs, for instance, and northern elephant seals, I think, they both had extreme bottlenecks, meaning that they were in like the few hundreds of members within, you know, the past 10,000 years or so, whatever it is. But what you can see, cheetahs, for instance, went through a genetic bottleneck about 12,000 years ago, and there were only a few hundred of them, only a few hundred, and they still today all display indications that they were reduced to a few hundred members 12,000 years ago. That's twice as old as the Earth is, or maybe just a little over as old as the Earth is. If
Well,
you
obviously, you're
10,000
using
years
the
old.
assumption that, you know, the Earth is, but I want to ask you, so a human genome or a human gene pool is around what, 20 to 25,000 genes deep. We're talking about an elephant population. How wide is their gene pool? So the humans, you know,
we can take
20, 25,000. What about an elephant?
We can take humans too if you want.
Okay, so let's look at humans now.
Noah and his wife passed now, or that already somehow also they also had huge numbers of alleles, and their children also had huge numbers of alleles. But regardless, they were.
Well, the DNA variety was in compressed, hidden form. But you're looking at humans today after the sorting out process has taken place.
Can you point
all I'm
to
saying is
where we see compressed DNA? Can you point that out to me?
Of course, we see that our DNA systems is now, you know, filled with overlapping codes. You deny the fact that, well, we're seeing that, you know, nucleotide sequences, which, you know, you can, and it's analogous to a sentence, actually has multiple meanings. You know, you can have a meaning frontwards and backwards. There's no book that, you know, you can find such. Obviously, one single cell is more complex than the space shuttle. A DNA molecule is more advanced than the space shuttle. We only speak about 1 to 3% of the DNA language, but you're trying to say that the vast majority of these non-protein coding genes are dysfunctional, or
well, we can show that they're non-functional.
Yeah, that's been shown. You only speak 1 to 3% of the DNA code and language. How can you
speak? We speak it. What does that mean?
Speak means understand. So we only understand.
We do actually understand a very large portion of our genome. That's how we know we have. I mean, obviously, we don't know everything. I'm not claiming we do know everything, but we do understand a large part of our genome, and that's how we know that so much of it doesn't do anything. I mean, where you bring up the ERVs that are involved in things like the placenta. Okay, we recognize that, so they're contained within the part that does have function. You look at something like gulo, for instance.
But it was always predicted to not have function.
No one predicts that it shouldn't have any function. That's not a prediction I've ever heard of.
Well, obviously, if these ERVs are functional, they're going to depend heavily on their position and their sequence. So if they're, you know, if they were positional and positioned even slightly wrong, or they had the wrong sequence,
so how does
functional? So, you know, that is more of, say, you know, an evidence and an indication of design versus say.
I've not heard. Well, for one thing, you have to then explain why chimps and humans have almost all of their ERVs in the exact same locations, along with gorillas and whatnot.
If these are plagiarized, no, I agree. If these are that's why the debate comes down to genetics and a genetic level, because if these plagiarized mistakes, if these AREs, ancient repetitive elements, if these are non-functional, that is evidence for universal common ancestry. But we predict that they are functional. That would show that God did place the DNA different. And I'm saying I don't understand how you can say that an embryological or placental or gene expression and regulation, how are these non-function? I mean,
I didn't say they were. I agree they are functional.
Well, how can they be functional if they're just evolutionary leftovers? That wasn't a prediction made by evolutionists.
I don't understand what you mean. I mean, we recognize not everything is a prediction, right? I mean, we recognize now that some ERVs do play roles. No one ever predicted.
Very important roles too, though.
Some of them, well, it depends. But I mean, yeah, some of them, you know, the ERVs involved in like the placenta. But no one ever predicted, as far as I'm aware, that somehow no ERVs should have any function. I've never heard anyone predict. And I have to ask, where did you get the idea that they did?
The thing is, based on these, you know, obviously EREs, AREs, you know, they are predicted in your model to be evolutionary leftovers.
Or where did you read that?
Aren't you? Well, I'd have to go look it up exactly. You know, are you saying that evolutionists have always said, oh, all this junk, you know, because you have the protein coding genes, which is what, 2% of our genome. The other 98% is junk.
Yeah, I'd say I think the protein coding genes are more like 10 or 11% or something like that.
Okay, so let's say 10 or 11. So let's say, the other 90 or 89%, from my understanding, they've always assumed that was junk. It had no function. It served no role.
Uh,
Am I
I don't
wrong on
think
that? Because I could probably look that up. I'm pretty
I
sure
don't know
I'm
if
right
they,
on that.
I don't know if they always assumed it was junk. I think that they figured a large part of it probably had to be, based on the way we understand.
Okay, let me ask you this then. Here's a
I
okay.
mean,
Just wait. Hey, let's just say these genes, which aid transcription in a fifth of the genome. Let's just say that they do serve regulatory roles. Obviously we do know. It sounds like we agree. You still say that they're non-functional evolutionary leftovers. If there's a mutation in that, say, nuclear DNA sequence, why would you say that's not deleterious? Because if it's a mutation in a functional gene, that will be deleterious ultimately.
how so?
Because
Even if it provides a selective advantage for the population?
well, at the end of the day, it's still going to be ultimately deleterious
How
because
so?
it's environmentally dependent. So if you're looking at
How could it not be environmentally dependent? How is it even possible for it to be anything other than environmentally dependent?
well, if we look at the genome and we say that it's polyfunctional, okay, because I'm trying to say there's overlapping codes, multiple meanings, multiple
It's
functions per nucleotide, that means it's polyconstrained. So even one small change
it's
in the
what?
genome will be detrimental and will be deleterious. But what you're not understanding is this is a prediction, a retrodiction, and an expectation from my model. So obviously you're
I've
not agreeing with it, but it's still consistent. Like I have no reason to give up my model. It's a competing explanation.
Well, for one thing, creationists are not publishing technical literature where they were predicting this long before anyone actually discovered it. This
I
is
think I think it was Michael Dent or not Michael Dent. Who's the one that wrote? No, obviously the Edge of Evolution was that Michael Behe? The Edge of Evolution or
you got
Michael
me,
Denton?
but the fact that
Well, he
large sections were non-functional was discovered long before uh, any of our modern intelligent designers or creationists were around. Probably most of the evolutionists too. I mean, this is something that goes way, way back.
But the thing is, now that we're seeing that they serve important functional roles, we're going to
It's
predict in the future. Like if you read "Replacing Darwin" by Dr. Nathaniel Jensen, he's predicting in the future. This is one of the predictions that we're going to see more and more functional roles based on these. And a lot of these silenced genes are actually because they play important roles in the embryo and in the placental
such
that now they
as
become silenced. Well,
such
even
as
the okay, like the ERVs, the endogenous
it's not
retroviruses,
silent though. I
and
don't know
other
what you mean by silenced. It silenced would mean it's not doing anything. If a
exactly.
mutation
And
is silent, it has no
there are some
effect.
genes that do nothing. Wouldn't you
The
agree?
ERV, the ERV that is involved in the placenta is not silent. It's doing
No, I
something.
no, I know. I'm just saying that the ERV that would be one of the functions. But we're saying that some of the nuclear DNA sequences that are assumed to be non-functional are actually functional in embryological development. But
I mean, sure, some of them might be. I'm willing to concede that. I think geneticists would concede. Any geneticist would say that also. They say, yeah,
well,
sure,
thank you.
probably some of it
And
is involved, but no
we're
one is.
predicting most of it because we haven't tested
But that
the
hasn't been
majority of the
borne out at
DNA.
all.
If we see this come to pass, this is consistent with our model of polyfunctional DNA, polyconstrained DNA, and very, very deleterious mutation load. That's my point. It's a prediction that we're waiting because if I believe we only speak about maybe 3% of the DNA language. We'd have to see what we
No,
actually do.
no,
I
no.
think. Are you saying that we understand the DNA language? Like if I put a kid in front of the hood of a car and say, "Tell me
Please
immediately what
don't,
you
please
believe is
don't use
non-functional."
analogies. Please don't use analogies. We went through. No, not we. We used a few analogies last time. I did a talk with Standing, or not Standing, with a bit of Orange. All he did was talk in analogies. I'm dead on analogies.
Well, I don't understand why an analogy say if a kid is ignorant of the function of a car and we're ignorant of the function of a DNA code. There's no. I know you don't like analogies, but that's a
But
very, very
geneticists
valid
aren't
analogy.
ignorant of the function of DNA. They've studied this very intensely.
And what are they finding out? Very, very
Very
significant
little of
regulatory
it is
roles. All
not very
very consistent
significant.
with our
Not a
model.
whole lot of it is devoted to regulatory roles. Not a whole lot of the and when they say functional, they
It's
include
transcripted.
things.
Yeah, it's
No, no, no,
transcripted.
no, 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 no. No, encode is what said functional means transcripted. That's not the normal definition. That's encode's very weird definition. What geneticists usually talk about when they mean functional is it's involved with
biochemical.
If it has biochemical function, it'll be involved in making proteins or involved in regulating the function of genes. That's what they mean usually. Yeah, the idea that something is functional because it's transcripted, that's weird. That's like, ugh.
Well, the thing is, where do you think these regulatory roles, these gene expression, because you have one gene that's helping another gene. I mean, there's all this interaction going on in our genetic information systems that we're just truly starting to understand the depth
such
of our
as
genetic information systems. Well, I want to go just back to the ERVs real quick. And I want to let you just answer this question. I'll let you talk. We know that there's cell processes obviously designed to weed out the harmful or viral infected genes, apoptosis. So how come after millions of years, why do we still have so many viral infected, endogenous retroviral cells? Because shouldn't apoptosis have ridden them long ago, but apparently it hasn't done anything.
Do you know what the lysogenic cycle is?
The lysogenic cycle?
Yes.
No, can you explain it?
So when viruses infect hosts, they go through one of two cycles. One is called the Lytic cycle, and the other is the Lysogenic cycle. The Lytic cycle is what happens when they enter the host and they use the host to start building their own molecular machinery so that they can break the cells and cause an infection, a viral infection. So they start building themselves. A lysogenic cycle is when they enter the host and they use reverse transcriptase to put part of their DNA into the host DNA. And the only way for that to work is for the host to not recognize that the viral DNA is in them, right? Otherwise, the cell's going to commit apoptosis, and then the virus is like, it's gone, right? So no, our apoptotic mechanisms are not by any stretch of the imagination perfect at weeding these sorts of things out, and that's why ERVs exist at all.
But the thing is, we see so many ERVs that apoptosis should have at least acted on a little bit. Because I don't believe that viruses are just going to randomly insert tens of thousands of, ERVs, which just happen to be in exactly, say, the right location and having the right, I said this earlier, having the right sequence to perform some valuable function. I mean, that seems like quite a big leap and quite a big, jump. Go ahead.
I'm a little confused by what you mean. What happens is you have these, so they use reverse transcriptase and they copy their DNA or RNA, whatever it is, into DNA that is put into our genes. And then if it's in a germline cell, then that's going to be passed on, right? Yep. It's going to be passed on to your offspring. So what that means is they manage to sneak past the defenses and insert themselves into us. And so what we have today, these ERVs that we share with chimps and with orangutans and all other primates, these are the very ancient fossils of those viruses. That's all that remains of them anymore. It's just these little genetic fossils. And so some of them have become incorporated into important functions after the fact.
Well, I think the fact that, they're being incorporated, because I don't think any of these endogenous retroviruses should have functions. So I don't believe that they're actually, plagiarized mistakes. And what I'm saying is, even based on those ERVs and based on the fact that I don't believe we speak much of the DNA code, them showing function shows that they cannot really be used, say, to build that, phylogenetic tree.
Well, actually they can, and here's why. Is that they're in the exact same locations. When you look at the synteny between chimps and us and gorillas and lemurs and all these other guys, and you can see that they're in the same places, that's definitely helpful for building a phylogenetic tree in the same way you look at.
Well, of course, we're living in the same environment. We're going to be, affected by the same.
No. What I mean is, when you pass down, when you have this germline mutation and you pass it down, your offspring are going to inherit it, and their offspring are going to inherit it if it persists, or if some mutation happens to cover it up, then it doesn't persist anymore. But let's just say it continues to persist, like in the apolipoprotein A1 family. The first guy got his mutation back in the 1700s, and so he passed it on to his kids, who passed it on to their kids, and their kids. And we can trace, because today we can trace all the people who have it in Italy back to this one guy in the distant past. And so that's the very basics of building a phylogeny, because you can look at the similarities that unite this little group right here of people, this little teeny group, and then you can look at all the similarities that unite, like native born Italian people or whatever.
Well, I get what you're saying. Obviously, you're using those DNA sequences, whether it's ERVs, whether it's nuclear DNA sequences, mitochondrial DNA sequences. But my whole point was that these phylogenetic classification methodologies, it's based on an assumption that common ancestry is true. But
if we
do see this accumulation of but the thing is, if we see a significant loss of information, loss of function, and genetic entropy, which I don't believe has been debunked yet,
it is. It's not taken
seriously. Is all it is an assumption.
So first of all, as you said, we can demonstrate common ancestry. You believe it up to an extent, like the kinds, the archetypes, or whatever, for instance. But genetic entropy isn't taken seriously by geneticists because it has failed.
Well, the thing is, I have many quotes from. I mean,
I can show you the data. I can show you I honestly, if someone says a quote about it, if I give you a quote, 100 quotes about people saying evolution is correct, you're not going to become an evolutionist.
Well,
likewise, if you send me 100
but you're trying to say that population geneticists don't take it
they
seriously.
Don't.
I mean, if you look at Alexei Kondrashov, he was the professor of evolutionary genetics at the University of Michigan. He said, why are we not extinct 100 times over because of these near neutral accumulation of deleterious mutations?
Who said that?
I think his name was Alexei Kondrashov. I don't know how to exactly say his last name. You've got Michael Lynch, who's a professor, population genetics. His quote was, "Our results provide no evidence for the existence of a threshold population size beyond which a population is completely invulnerable to a mutational meltdown, mutation accumulation, and the extinction of small populations." The thing is, they have to hold to their worldview, their primary axiom.
No, they don't.
It's a secret among population geneticists that this is a major and detrimental problem.
It's not, though.
I'm giving you quotes.
It's actually not, because, and here's why. Again, quotes really don't do anything, because we can show
quotes from population geneticists, evolutionary.
Have you actually read the technical paper on it?
I sat and read that letters to a creationist, and none of those arguments were very important. I want to hear your best argument against these accumulation of near neutral mutations that's degenerating our information system.
Actually, geneticists have already debunked it, because for one thing, creationists ignore beneficial mutations, which offset.
I've already told you that these beneficial mutations are rare, so natural selection may amplify the beneficial mutations and get rid of the, most, because obviously natural selection is acting on the phenotype. It's not going to recognize one single point mutation on a nucleotide. It's got to act on the phenotype as a whole. So at the end of the day, those near neutral mutations are not selected against. It doesn't matter about beneficial mutations. It's a straw man argument. It proves that Scott guy in that letter to creationists, he didn't even read anything that John Sanford provided. Because
I'm sorry, you're going to have to say that again. You're saying a whole lot about you were saying a whole lot at one time. For instance, let's break this concept down.
Well, let's talk about beneficial mutations. You're saying that beneficial mutations are going to counteract these accumulation of near neutral mutations, but
which has been shown. Even the model that that Sanford was basing his model on was oh crap, what was his name? Kimura. Kimura's model. He was ignoring that Kimura had pointed out that beneficial mutations would offset the harmful and near neutral ones.
The near neutral mutations are not selected for or against, because you're going to have to show me thousands of these examples of adaptation via beneficial point mutation.
Why?
Well, of course, because you even admitted that the overwhelming amount of mutations are near neutral. If you're saying that beneficial mutations are going to help with this, where are the beneficial mutations in man? Because, Jackson, it is very well documented that there are thousands of very deleterious Mendelian mutations accumulating in the human gene pool, even though there's strong selection against such mutations. So where's all the beneficial mutations in man?
I mean, I've provided you but one already, which is the apolipoprotein A1, which is beneficial. It's beneficial in the sense that it provides a selective
Loss of
advantage.
specificity, though.
What is okay, so when you said specificity, what we're talking about is this a thing that's now better at its job than it used to be? I don't know what you mean when you say it's lost specificity. It's now better
because the fact, well, I'll explain it to you.
It's better at metabolizing cholesterol than it used to be.
No, because
that's what this thing already did. This protein already did that job.
Well, do you want me to explain what I mean by loss of specificity? I'm saying that the fact that the person with the mutation is unable to produce the normal levels of HDLs, they're known, and you probably know this too, they're known to perform a valuable role in moving bad cholesterol. This suggests that there could be a health downside to this mutation. It's exactly like sickle cell anemia. It's exactly like the HIV example. It is.
Yes, apolipoprotein A1 is not like that at all. It's not an example of heterozygotic advantage. I have not seen any technical literature to indicate that that is the case.
Well, the thing is, you can give me one or two. Obviously, we're going to agree to disagree on that one. You know, I just showed you why it's a loss of specificity. But where are these thousands of examples of, because we document thousands of deleterious Mendelian mutations, but we struggle for 2 hours to even agree on one solid beneficial mutation.
So here's what I defined what a beneficial mutation is. It provides a selective advantage. And in this case, and it doesn't matter whether the gene, it doesn't matter what has happened to the gene per se. It is providing it underwent a mutation that now provides a selective advantage.
That's still deleterious in another. If it's polyfunctional and polyconstrained, even the smallest change is deleterious.
What does that mean? What does polyfunctional and polyconstrained mean?
Poly meaning many, functional meaning, you know, performing many functionals in even a single nucleotide.
So how is the apolipoprotein A1 polyfunctional?
I'm saying that our genome. I'm saying that our genome is polyfunctional.
So the whole genome is doing multiple functions?
Our genome is performing multiple functions. One gene is regulating another gene. I mean, there's overlapping codes. It's the double helix. They're all interacting with each other.
Whoa, what? That's not what Okay. The regulatory genes are doing specific jobs. You have one job. I mean, they might not necessarily have one job, but they have multiple jobs, polyfunctional. The point of their job is, at least one of them, is to regulate other genes. Okay, that's their job. I don't understand what you're trying to get at, because
I'm trying to say if the majority of our gene sequences are proven and shown to be functional, any change in our genome is going to be deleterious. That's what I'm trying to say.
Okay, so and here's why I explain that it's not. Because you have examples.
I know you think you've explained that, you know, they don't perform roles, but I think it's pretty evident that they do perform roles, because
because you haven't provided examples that they have. You've pointed me to these ERVs over and over, which we've already agreed. The few that actually have any job have jobs. I'm totally willing to concede that point, but the majority of them don't have any jobs.
Well, do the majority of Okay, but
ERVs don't have any
of the deleterious mutations, are they going to display any clear phenotype? Like, let's say it's just a neutral mutation. Is it going to display any clear phenotype?
Not necessarily, no.
Okay, so natural selection acts on the phenotype as a whole. It doesn't act, you know, on a genomic level
it does.
Per se. Well, it's looking. We're all mutant. You know, one maybe more mutant than the other, but natural selection does act on the phenotype.
Yeah, I mean, it acts on the phenotype and the genotype, yes, or genome.
Okay, so how come there's no beneficial, you know, birth anomalies being seen? You know, how come we see so many birth defects, but we're not seeing any, you know, positive outward phenotypic beneficial mutations that are as evident as these birth defects?
So, for instance, when I was referring to Ragavalia, which have duplicated these genes, they now have novel propeller fans, which allowed them to invade a new environment. Is that or is that not beneficial to them? Is that not selectively beneficial?
I'm not crazy familiar with that. So even if I concede that, say, because to be honest with you, every example that I've seen an evolutionist provide, it has, through further research and looking into literature, provided sufficiently some type of deleterious effect. Do you believe that that type of example is sufficient to say prove pond scum to people evolution? I mean, is that type?
No, that wasn't my point at all to do that. I was merely pointing out the existence of beneficial mutations. I'm not even talking about evolutionary history. I'm merely explaining how the process of evolution itself works.
Okay, if I concede that that's, you know, a beneficial
Well, I can
mutation,
move on.
there's
We
no
can go on
deleterious
to another
effect.
one then.
Okay, I'll just let's just say it is. The thing is, you know, I think I might have said this before. You need to understand that obviously a population it's going to be undergoing genetic decline due to the vast numbers of slightly deleterious mutations
No,
that I'm talking about, because selection
we
can
don't see that
amplify this
actually.
beneficial mutation. Well, you're saying selection's amplifying this beneficial mutation, right? It's amplifying it now, and it's
In what?
in the one you just talked about with the.
Well,
Was
no,
it
it's not amplifying that. It's created this novel structure from these genes. The same with the notothenioid fish, was that you had a gene that had a totally different function that was duplicated. It developed a new protein and a new function.
But is that going to stop the degeneration process
There's
ultimately?
no degeneration process that's been observed. That's not
Okay,
being
so
observed in actual real world populations.
okay, so you don't believe that these near neutral mutations are degenerating our genome, right?
I didn't say I didn't. I didn't say I didn't agree with that. They probably are. But what I'm saying is that the genetic entropy that you're
I'm just
talking
saying
about.
there's nothing that can stop it, much like nothing can stop a rust on a car. Sorry, I had to use an analogy
No,
there.
that's no. That, I mean, that's fine. It's. I'm not saying that near neutral mutations aren't occurring and accruing in the genome. That's not what I'm saying at all. What
Okay.
I'm saying is that the magnitude of which these occurring is not causing genetic entropy, as Sanford would like us to believe, and as other creationists would like us to believe. That's not happening. It's not being observed in populations.
So you don't think that there's any, like, for example, RNA viruses, it's if you look into the Spanish flu, H1N1, you know, they killed what, between 50 and 100 million people. By 2009, it was extinct after 15% of its genome of 12,600 nucleotides had mutated. So it actually, and if you look at the graphs in Genetic Entropy, the book, it followed a perfect linear mutation accumulation rate and a perfect exponential biological decay curve. So that means the H2N2 and H3N2 outbreaks are now following the same decay curve. So
Why
we're
don't we
seeing
see
it
that in
in real
anything
time now.
else?
We're seeing it in real time.
Why don't we see that in anything else? I honestly, I'm not familiar with those examples, and I'm very leery of anything Sanford says because he's.
I'll send them to you.
But is anyone else aware of those examples?
Well, like I said, I've given you quotes from.
But have you yourself read the technical papers on the examples he provided?
Okay, well, look up James Crow. He's another. I've already given you 3 quotes from population geneticists.
I don't care about the quotes.
I
The
just want to
quotes
give
are
one for the
irrelevant.
audience. I want them to hear this one. So he said, I do regard mutation accumulation as a problem. It is something like the population bomb, but it has a much longer fuse. So these evolutionary population geneticists, they are recognizing it as a problem, but they can't concede it the way we do because they have to hold to their primary axiom. They have to hold to molecules.
In fact, I would say the exact opposite, because what I see in researchers is that they are honestly struggling with the actual data that we see. And I can turn that around and say, look at Todd Wood. He said evolution is definitely true, but that he rejects it because he wants to believe in the Bible. Well, and he's not the only one. Kurt Wise has said the same thing. And I could go on. We could look at other examples. So I don't care about quotes. I really don't. They don't mean anything.
Well, because at the end of the day, just like Todd Wood, Dr. Nathaniel Jensen, they recognize the threat of these, you know, ancient repetitive elements, of these pseudogenes, of these non-functional region codes. But because, yeah, you can use those plagiarized mistakes. It's like almost like a teacher, and I'm going to play devil's advocate here. If they catch, you know, one paper and all of a sudden another paper has exactly the same answers, but also exactly the same mistakes, you're going to conclude that they copied from somebody, right?
Oh, what is this? Scott disapproves Sanford. I think it's the quote. The quote of Crow is a quote mine. I'm not entirely surprised.
Actually, Jackson, talk to your audience for maybe 30 seconds. I just got to run upstairs. My 6-week-old is crying. Just 1 minute.
Okay, good. Okay, Nestle, let me take a look at this. Thank you for that. Let's see. Holy crap, this is very, very long. I suspect standing is just a quote minor. I wouldn't be surprised. Scott, just okay. So let me see. Let me look up Crow speculation. Blah blah blah. Repeatedly refers to a 1997 article by James Crow, in which Crow speculates that human genetic fitness may be deteriorating by 1 to 2% each year. This sounds dire, but Crow makes it clear that the only reason he makes this suggestion is that modern hygiene and small family sizes have largely shut down natural selection among humans, such that much of the world is now running a gigantic mutation accumulation experiment. In a 2010 article, Michael Lynch derived a similar estimate, 1 to 5%, of the expected fitness decline per generation for humans in industrial societies where improved nutrition and medical care allow the vast majority of individuals to survive to reproductive age. All would agree that under those circumstances, deleterious mutations will accumulate and fitness will decline at some rate. Crow picked 1 to 2% simply by analogy with Mukai's fruit fly studies. Crow makes it clear that this fitness decline is merely a speculation on his part, not based on evidence of actual fitness decline, and that this proposed genetic deterioration only pertains to the last few centuries, with greatly reduced rates of infant and childhood mortality, not to mankind's history in general. Very interesting. I will try to bring that up. Thank you, Nestle. So I would pay you the big bucks. So, Crow is saying that populations that are not subject to selection, fitness goes down. This is in the absence of selection. Sanford's claim requires that fitness go down despite selection. Interesting.
Hey Jackson, my man, I'm back. Did you?
Okay, well we
I probably got another little bit, unless the baby cries. Hey, did you want? We can continue with this. Did you want to go into? Because I know you wanted to talk about human chromosome 2, because that's a big challenge.
I do want to point out, since you're back, we looked it up, or Nestle looked it up for us. Thanks again, Nestle. That the quote of James Crow is a quote mine. Actually, he said Sanford is misapplying his quote. What he's actually saying is that populations that aren't subject to Crow is saying that populations that are not subject to selection, fitness goes down, but this is in the absence of selection. Sanford's claims require that fitness goes down despite selection.
Yeah, but I'm trying to show you here that natural
entirely
selection
opposite to what he's claiming.
No, he's recognizing that there is this issue, but obviously he's invoking natural selection. Of course, he's got to have a rescue device. I mean, what's your rescue device for these homologous structures that don't entirely fit within the evolutionary picture? Convergent evolution. Like convergent evolution. Well, for example,
don't call them homologous. I mean, that's like okay, so first of all, I think you should point out, because it's pretty obvious at this point, that Stanford quote mined the guy, because we just read the piece on it, and I can put it in the description after the video.
Is this James Crow's?
This is so the guy. So this is I don't know who this. This says letters to creationist, and he's talking about and he's citing the Crow papers and talking about James Crow's speculation on human genetic deterioration. I just read a paragraph to the audience. And so Sanford is quote mining the guy. What he's saying is actually completely opposite.
Well, he does indicate that human fitness is declining 3 to 5% per generation.
Okay, in the absence of selection. Sanford is saying that it's deteriorating while undergoing selection. So they're completely opposite claims.
Well, he's just saying that the rate of human degeneration would be slower based on the fact that natural selection is going to.
That's not what he's saying, though.
Yeah, but Archie, isn't the reason why you don't believe genetic entropy and Crow and all these population geneticists don't believe that?
I disagree with the population geneticists. I mean, this guy is clearly getting quote mined. This poor guy's saying one thing, and Sanford's saying that is quote mining him as saying another. It's like the Darwin I quote.
Well, no, because he's admitting to the current human genetic degeneration, but he's saying that it's not going to, go as rapidly as
absence of selection in a post-industrial age.
Exactly. The whole debate the whole debate is regarding, is natural selection able to stop these accumulation of near neutral mutations. And I don't believe for a minute that it can, because just like a car, and I'm going to go an analogy here, I can change the brakes, I can change the windshields, but at the end of the day, those rust molecules are still going to build up on the metal no matter what. Nothing's going to stop the degeneration.
Crow's talking about buses and Sanford's talking about airplanes and saying they're the same thing. That's in essence what's going on here. It's totally this is a pretty clear quote mine. Okay, I don't know how to make it. I mean I can see it right here, and he even cites the articles.
Is this from the letters? Is this from the letters to creationist?
Yeah, it is. And so the guy is citing the actual papers where Sanford is invoking, and so it's pretty clear this is a quote mine.
Well, let's just say for argument's sake it's a quote mine. It still doesn't detract from the facts that natural selection clearly is not going to get rid of these near neutral mutations. I think it's pretty obvious that these deleterious mutations are accumulating.
It's not
even with natural selection, which obviously we agree to disagree, but I think it's clear evidence of biblical creation. That's exactly what we predict is a decay process and a degeneration process in the universe and on a genomic level.
So when we look, like I said, when we look at actual populations, this isn't occurring. I'm gonna really want to look at the example you provided, because I imagine that it's not exactly what's going on, which tends to be the case. But I'll be intrigued to look at it. I will definitely look at it.
Look at it.
But I mean, what I was saying is that this isn't occurring, or this isn't being observed as occurring in normal populations as far as population geneticists are concerned, or can see. And so I know, because I actually worked in the Department of Zoology, and none of the zoologists were ever indicating that genetic entropy was actually happening. But
the thing is, okay, let's just say that there was an experiment. Okay, let's just say we're looking at an experiment. Would you expect that there would be clear evidence in an experiment? Because the whole point of it is the overwhelming majority of mutations, they're very subtle. They're invisible, right? That's what I've been saying, near neutral. So they're not going to express a clear phenotype. So that means in these experiments, let's say we were to look up some, almost all mutations are going to be invisible in these types of, because they're only monitoring gross differences in performance. So like the high impact mutations, they're obviously going to be observed. But the high impact deleterious mutations, they're still going to be selected away in such experiments. So,
I mean, I don't know what you mean by high impact. There are lots of mutations that have low impact that are still selected for or against. That still happens.
Well, yeah, but it's that accumulating, invisible to natural selection, the near neutral mutations that are the issue. Obviously, we're just gonna have to, agree to disagree on that. My whole point is, this is consistent with biblical creation. I have no reason to abandon the biblical model, because to me it seems pretty clear that there's an overwhelming amount of deleterious mutations versus beneficial, because we can agree there's thousands of deleterious mutations versus beneficial. Go ahead.
According to your model, and this is something we were talking about earlier, but in the real world is precisely the opposite of genetic entropy. If genetic entropy was going on from the very beginning,
well, we can see that there's so many more diseases, genetic diseases.
No, it's not about it's not about that. It's go back to our elephants. Earlier we were talking about, they have no way to transmit the huge numbers of alleles that they magically inherited to their offspring. They have no way to accomplish this in a way that would allow it to propagate and spread and accumulate, accumulate mutations and have more alleles in the future. There's no way for that to occur unless they are, again, mutating orders of magnitude higher than anything we observe. And we know this because we observe how genes are passed down through populations.
Well, we can see how quickly. We can see how quickly. There's over 350 known breeds of dogs, and we can see just in how many years that these dogs have been artificially bred from.
Okay, but no one's artificially breeding elephants.
Mongrels.
No one's artificially breeding elephants.
Yeah, but I'm just. Are you recognizing? I just want to make sure you're not misrepresenting our model. You do understand that we believe 90% of the nuclear DNA differences are not the result of mutations, though, right? You reckon you're not strawmanning me, are you? Like you know, we believe that that those are based on
90% of the differences. I don't know what you mean.
Well, I've been saying this the whole time. We believe that these tens of millions of nuclear DNA differences, they're not based on
Between what?
mutation. Well, like these non-functional DNA, non-protein coding regions, we believe they have function. We believe that these were placed into each created kind. That's what allowed for such variation, such speciation, because it's been a sorting out of information. I just want to make sure that you at least, I understand you think it's wrong, but you understand what I mean
I can
by
show that it's wrong.
Okay, if you want to say that this is far-fetched, how can you go from a single cell to a multi-cell to a fish to an amphibian to a reptile to a mammal to a bird to a human by a vast amount of deleterious mutations? I mean, where's the novel? I understand you've provided a couple examples, but is that enough for you to sufficiently conclude that this uphill evolutionary process occurred?
That's not uphill. Well, first of all, I think saying it's uphill is a complete misunderstanding of what's actually going on. There is no uphill in evolution ever at any point because there is no direction in evolution.
So at one point we had a single cell, bacteria, and billions of years later we have whales and elephants and pine trees and humans. Obviously, there's a bit of an uphill change there.
There's not. Actually, no, there is no uphill. There's no such thing as uphill in evolution.
Does a bacteria have a working gene, say for like a heart or a lung or a fin?
It doesn't have to. Well, I think that's a fundamental misunderstanding that a lot of people have about evolution, is that it doesn't care. Evolution does not give two craps whether you have one heart, three hearts, or no hearts. As long as if you are surviving and passing on your genes, that's what it cares about.
If a population of bacteria, if a population of bacteria is passing on its genes to more population of bacteria, you're never going to get a gene for a working. You're never going to see that huge large-scale change that you want us to believe in.
We do have there are actually very good models of how hearts first developed not in bacteria but in early metazoans.
Well, I'm sure that there's a lot of fancy stories
it's not stories, it's actually stuff we can test.
I don't believe it can be demonstrated. The origin of so are you saying that an organism that didn't have the information for a heart, there's a working...
What is information?
Do you want my definition for information?
Yes.
So a definition of biological, that it's not easy to come by, but if I had to give you a technical definition, I'd say an encoded, symbolically represented message conveying expected action and intended purpose.
Okay, so that made it about 10 times more complicated than it already was.
Exactly. So let's make it easy.
It's not complicated
for the audience. Okay.
What I mean is it's not complicated because it's technical. Precisely the opposite. It's entirely subjective. It seems the entire definition does not have a correspondence with what's actually happening. It doesn't. The concept of symbolism in DNA doesn't make any sense. There's no symbolism in DNA. DNA is a molecule that's doing a set of jobs. There is no expected action.
But there's very specific genes that are coding certain, whether it's organs, tissues. I mean, the information system of the DNA code of a bacteria is far different than the DNA code of a whale or a human being.
I would agree. I would absolutely agree with that.
Well, exactly. So I'm trying to say, how do you
there's no uphill. There's no uphill there. There's only these changes that are occurring when what we understand about the acquisition of mitochondria, and the origin of eukaryotes, is that this was just a combination of two prokaryotes, right? This had nothing to do. There was no uphill thing that was going on. This was all about these organisms that were co-opted and into different functions. You have one that became, and depending on whether it's the, from the nucleus out or the nucleus on the inside, whichever hypothesis you tend to take, it's still, there's no uphill there. There's still no uphill. There's nothing.
I'm trying to say, if the vast majority of, because for example, and you've done a good job to at least avoid very obvious ones, but when evolutionists cite examples, say of, whether it's new information, new traits, they're invariably citing whether it's evidence of new traits, but they're always caused by the corruption of existing information.
Well, do you know what they are referring to when they say information?
And they say well, I mean, I like to
go
use
and sit in on a freshman biology class in college, and the professor says the genome has information in it. I know what he's talking about because he uses very precise language to describe it, and he is not going to use terms like symbolism or expected function. What he's referring to is he's talking about protein coding genes.
The protein coding.
So
the DNA,
if you say that's information, and we have genes, we have duplicated these genes and created new genes with new functions.
So you're saying mutations, they can create new varieties of old genes. So whether it's in
we have observed that, yes,
light-coded lab mice, but let's say damaging mutations, typographical errors. Do you believe that that's vindication for molecules to people evolution? Like, you believe that that change can take a single-celled organism indirectly, obviously it didn't go into all the life we see today. I mean, you believe that, given
there's a difference between I think we might be talking past each other. There is a major difference between just what the process of evolution is and what the history of life on Earth is. We have to establish that there is a process by which this, the process was discovered before people had put all the pieces together. I mean, they already the geologists, the naturalists had a very extremely vague picture of what may or may not have happened in earlier times. There was an age of fish, an age of reptiles, an age of mammals. That was pretty much it. Darwin proposed a mechanism by which species could originate which was through natural selection, and then of course we've added more mechanisms, genetic drift, sexual selection, etc. in the years.
And Darwin, and obviously Darwin didn't know, nearly as much about, the genome as we do now. I mean, he just thought that the cell was like a glob of. Obviously, it's a totally different story to that.
No, absolutely, I agree. Yeah, and that was a result of the the early 1900s, early to mid 1900s, and so of course we've absolutely learned way more. Darwin would be blown away by the amount of information.
But do you believe that Darwin would believe that these typographical errors, these mutations, are the answer to the large-scale evolution? Like, do you believe that he would be satisfied with that? Because he just believed natural selection.
Be satisfied with mutations occurring? Because I mean, in his day, he understood that there were variations in organisms, and these variations were beneficial or deleterious, and if they were beneficial, they were inherited; if deleterious, not. And so that's what ultimately, if there were speciation events, caused new species. And so that's the whole of evolution. And then when you apply that process to the history of life, we can see how that's occurring. We can see how this history of life is unfolding.
Well, I mean, you can't really see
that process.
Obviously, if you're looking at
in some cases, we can literally. For instance, in foraminifera and radiolarians and coccolithophores, we can literally see these speciation events happening in the fossil record because of how well things like forams get preserved. So yeah, you can actually see their very, very nice slow progression or fast progression.
But at the end of the day, like we said before, if all the 350 known breeds of dogs went extinct today and we dug them up, we could arrange them in any order we want. We can give any interpretation we want. That's why I say the debate is solely on and in the genetic realm, because fossils, they're not exactly, obviously fossils aren't inherited. Genes are inherited. Traits are inherited. That's why the debate, I don't think, the fossil record is evidence for creation or large-scale evolution.
I disagree very heavily, and that's why part of the reason is, I've devoted an entire series talking about paleontology, but it's when we look at the fossil record, what we see are instances of mass extinctions followed by periods of bursts of biodiversity, of radiation to different groups of organisms. That's what we see throughout, for instance, where we have.
Well, we see billions of dead things obviously buried in sedimentary
You
layers.
understand that we can tell a lot from fossils, right?
Yeah, but all I'm saying is if all the known penguins today, went extinct, and then man 1,000 years later, dug them up, they're going to conclude that it's some big transitionary fossil, or they're going to have some interpretation that's probably nowhere near the accurate.
How do you know that? Can you actually provide me any evidence that has occurred?
Well, you agreed last time that there would be various interpretations based on, the 350 known breeds of dogs. If those were dug up, you said that, there'd be conflicting interpretations.
I mean, if you look at the teeth and they all have very similar teeth, then you could probably imply a good degree of phenotypic plasticity among this group, which is why teeth are so important to mammalogists.
Well, there's only going to be so much you can get from the bones. That's why I'm saying that the debate is about genetics.
I mean, that's why
what's inherited.
The fossil, I agree, genetics are a big part of it, but fossils are also equally very important because
I believe that. No, I agree that fossils are important to an extent because it's all based on an interpretation based on whatever our initial
for instance, assumption. What we don't see in the fossil record is any indication of a creation event or a flood event. We don't see that.
You don't believe that all these, because obviously, fossilization, it requires rapid burial. So I mean, if we
not
see
necessarily by water
of dead things, well, I mean, we don't see very much fossilization going on. But my point in the fossils, though, is
okay. Wait a minute. Wait. When you say we don't see very much fossilization going on,
we don't see very much fossilization going on today.
How have you determined that?
Well, for example,
how do we determine what a fossil is? First of all.
Well, if it's buried rapidly and well, are you trying to say that we do see significant fossilization today?
Okay, how would I determine today what is going to be a fossil? I can't do that. Fossils are only determined as fossils after the fact.
Well, because let's say a whole population of buffalo dies in, some.
If you bury a dead fish, then it may fossilize or it may not. I can't know for sure. I mean, bioturbation by worms or whatever might destroy the bones, and so it might never get fossilized. I don't know. Yeah. We can't tell in real time.
Yeah, but how can we find these fossilized fish, in the middle of birth or in the middle of eating each other? We see these clams that are fossilized in the closed positions. Normally they fossilize, in the open position because their muscles relax. I mean, there's obviously evidence, because you said there's no evidence of a global flood, but if you have the assumption that there was a global flood, I mean, we see it all around us in the sedimentary layers. We see it in the fault lines.
Here's the problem: is you have to assume it happened and then reinterpret the evidence accordingly. That didn't happen for evolution. They found a good number of fossils. Darwin was, for instance, a fossil hunter, and he went and found fossils, for instance, in South America, and he realized these fossils that he's finding share a whole lot of commonalities with the organisms that live there currently. Of course, he was finding mostly like Cenozoic organisms, things that had died within the past few 100,000 years or so for the most part.
Yeah, but you're basing all these fossils because you're building your phylogenetic tree, and it's all fitting neatly into a nested hierarchy, right? Because that's
we can look at that. That's to the 5-year-old from earlier. We could sit a 5-year-old down and say which of these is not like the other, with a lion, a human, an elephant, and a jellyfish, and they're going to say the lion, human.
Well, I'm trying to say that there's a competing model and explanation that would also predict a nested hierarchy, because creationists would predict a nested hierarchy.
Why?
That's why. Well, I'm going to use an analogy. Do you want me to use it, or do you want me to?
I want you to tell me specifically using data why you would predict a nested hierarchy within creationism.
I'm trying to say that based on design and based on the fact that God created us in his image, there's evidence there that, we're going to be able to see things that we design.
Did he create elephants?
Correlate and infer that that's the type of way that he might have. Because if I actually, and I'm going to use this, I know you don't like analogies, but for the sake of argument, I would say the means of transportation naturally fall into a hierarchical classification scheme, because we're going to recognize one category that we call cars. And even though each car, Jackson, is unique, you're going to have to agree that all cars share a set of features.
Can we go back to the drawing boards at any point and completely redesign cars totally unlike anything that's ever been designed so far?
Actually, there has been a descent of modification. Like if you look at a 2018 Cruise and you look at a Cruise 10 years ago, there's been modification. You can almost say that they've evolved.
Can we completely? No, I wouldn't absolutely not say they have evolved. At least not in the biological sense.
Oh, obviously, yeah, in the change sense. But I'm trying to say, just wait. If we shrunk, just like in biological life, if we shrunk the list of shared features, say in pickup trucks, SUVs, and brought it into the same group with cars, we're going to be able to see slowly that nested hierarchy building. And the more vehicles that we try to, say, lump into our transportation classification system, the smaller, just like in the biological realm, the smaller the list of shared features becomes. Remember, and it's just like what we observe in the Linnaean classification system. Go ahead.
Okay, remember how last time I pointed out that cars don't reproduce, therefore they don't pass on inherited traits?
Yeah, but that's a straw man with our model, because our inference is based on design. Why it's not.
Because what you can't, and here's why analogies do not work in this arena. Because
I just want to, because you didn't let me respond to the reproduction thing last time. You kind of just talked over me. I think that's a bad example, because I'd like to see how reproduction came about anyway, since it's extremely complex.
That's a non sequitur.
And secondly, I think that the evidence of design and modes of transportation, lines of code, say in Microsoft Excel, PowerPoint, and Microsoft Word, I think it's so evident of a common designer that the only explanation and rebuttal that an evolutionist can give is, oh, well, they don't reproduce. I think completely
it's not.
That is unconvincing, and I think it's a straw man. Go ahead.
I can give you another one. Actually, we can't place cars in nested hierarchies. We can't do it because we can't actually, because different manufacturers can. No, actually, we can't, because manufacturers can borrow designs, and if two manufacturers borrow the same design, the exact same design, they are both creating the same thing, then the nested hierarchy falls apart, because this is the same structure on two different unrelated things. For one thing. Second of all, no, the reproduction thing is actually is the whole reason evolution happens is alleles are varying as they're being passed on generations. That's literally what the definition of evolution is.
I agree that evolution,
and so cars
frequency and populations over time.
Biologic evolution. So cars, that analogy doesn't work in any respect. And so you can't place cars in nested hierarchies. What we can do is we can place. No, you didn't, because when two different manufacturers, you're talking about two different clades of cars, share designs, they have just crossed. How do you plot that on a phylogenetic?
Okay, so you're saying Chevrolet and, say, Chrysler. Okay, so they're two totally different designers, two totally different manufacturers. They're borrowing design. I mean, I don't understand how that's an argument against. Because if we look at cars, they occupy, say, the genus and species level. Trucks, SUVs, and cars form a family. Orders represent the different, say, purposes for land-based vehicles. And you could just go on and on and on. The environments in which transportation operate, that could constitute as the class level, say boats, airplanes.
The manufacturers can at any time go back to the starting point and redesign a car totally unlike anything ever, anywhere else ever.
Are you saying like making a whole new car?
Yeah, you could go back and make a 5-wheeled car or something that's totally different from any other car. You can't do that sort of thing in biology. It's not possible. What
exactly,
we see
all this variation that we're seeing in species is all based on pre-existing genes.
And I'd like to see a paper demonstrating that. I'm still waiting from last time, because it doesn't happen. Because that sort of thing does not happen. It's not. There is no just simple degradation of mutations that's occurring, because that's not being observed in the population.
I'm showing you that there is a competing model. Obviously, I understand you don't agree that the the modes of transportation actually fit and show an example of common design, but that's why I personally believe it comes down to a genetics level, because the fossil record can be interpreted through a creationist lens and an evolutionary lens.
I've looked at it through the creationist lens, and
it's genetics where the game is won, and I believe that it's being won. On the creationist side. Go ahead.
And so here's why it's not, because Sanford, his model is not accepted among actual geneticists. And you can ask the actual geneticists. You don't have to just listen to these quotes that are being given by other creationists. I encourage you to actually go ask the geneticists, read their papers on this stuff. The models, like Sanford's model, are not accepted by anyone outside of creationism. When we actually look at what the data shows, is that you have new structures and functions and proteins which originate through completely natural means, and I provided examples. This is not an indication of Sanford's model. It's precisely the opposite, and so that's why it's not only not accepted. It's not a model that's working. It's been tested. That sort of thing has been worked out already among the geneticists in the field. They've already worked this stuff out. It doesn't work.
Okay, so I would say our model predicts, and I understand everything you're saying. I would say, and we'd have to test this and demonstrate it, so that, that would be the burden on us. I would say, we're going to predict limits. We're going to predict that our genome is functional based on design. We're going to predict, the majority of mutations are very deleterious. A lot of the traits, the, gains of functions, the changes and variations we see are mostly due to a sorting out and a loss of information. While I believe that, God originally wrote the genetic script, we're now seeing variations based on the genetic script. I don't believe mutations are a genetic script writer. I just believe they're merely typographical, obviously, alterations on a genetic script that's already been written. So I think that's where the debate comes down to. I think we've had a good conversation. I'll allow you to give a few, closing words.
Oh, we got to go.
I won't interrupt you. And how long have we been going for? That's because
heck if I know.
Hour and a half.
Oh, something like that. Yeah, for thereabouts probably.
I'm gonna let my baby cry for 2 more minutes, because I'm curious with human chromosome 2. Okay, now obviously, if humans and chimps, and tell me if I'm getting this right, if they share, let's say they do share a common ancestor, Jackson, then that means we should observe a fusion event, right? So what do we see? We see human chromosome 2. It is showing evidence of a fusion event. Therefore, humans and chimps share a common ancestor. But isn't that's
not the one
logical fallacy of affirming
necessarily? No, not exactly. I mean, that's in science. In the enterprise of science, if you make a prediction and then you find evidence that fulfills that prediction, if you say, aha, this evidence fulfills my prediction, that's not a fallacy. I mean, that's not how science works. It's called making a prediction. This isn't like
what would have happened if they discovered that humans and apes both have, or chimpanzees, both have 46 chromosomes? Would that be evidence for evolution too?
Would it? I mean, what happened was they made a prediction. They'd already what the really good thing, or the really good evidence of evolution, is that there's already this huge amount of synteny between humans and chimps. That's already a really good indication for evolution, because we understand how, like I said, traits are passed down. So, but what happened with the chromosome was you have, these researchers actually had to try to find a paper, because some creationist was asking me a while back to find like the original paper that predicted the fusion event, which is really hard because there likely isn't one. Well, it seems a number of it seems like a number of researchers surmised before we could actually look directly at the nucleotides that there was a fusion event based on, for instance, how long chromosome 2 was. But they predicted that if we share a common ancestor with apes, or with the other apes who have, the 48 chromosomes, then we should find a fusion event indicating that these chromosomes fused, because they couldn't just drop off the earth and disappear. That would have been lethal.
Was this after? I just want to understand. Was this after they discovered that humans have 46 chromosomes and chimpanzees have 48?
Oh yeah.
So they realized that. Okay, so you're saying they realized that there was a difference in chromosomes. So now they, went to the drawing board and made this prediction that there must be a chromosomal fusion of some kind.
They knew about the karyotypes since long before they actually found the fusion event. Yeah, we've known about that for much longer. But But yes, that's essentially what happened. They predicted because they saw we're missing a pair. What the heck happened to it? And so they surmised that based on the size of 2 that it probably contained a fusion event. And so geneticists were already figuring this out, and then we could look at the actual nucleotides and whatnot. We can realize, oh hey, actually there it is. There's the vestigial telomere. There are the vestigial centromeres, and so that's in essence what happened.
And correct me if I'm wrong here. Doesn't the human chromosome 2, doesn't it have like 150,000 more genetic letters than the chimp chromosome, say 2A and 2B?
I'm sorry, say again.
Does the human chromosome 2 have 150,000 more genetic letters than the chimp chromosome 2A and 2B? Sorry.
I mean, I believe it is highly.
There's a huge difference.
It's somewhat degenerate, and that was one of the things Jeffrey Tompkins was kind of whining about. He was like, "Oh, it's not degenerate enough."
Well, how come the fusion, say from an ape, why would it? And I'm not trying to be argued. I just want to why would it result in the extra DNA of 150,000 more genetic letters? I mean, that seems odd.
I mean, insertion events can insert large numbers of nucleotides. That's not unusual at all.
Was this after the human chromosomal fusion?
I haven't looked into that part specifically. What Nesslig and Rational Mind and I looked at was demonstrating that it is indeed a fusion event.
Okay, let's just say for argument's sake it is.
Which I've actually met other creationists who would also accept that it's a fusion event.
I would probably say that there's strong evidence it is. So let's just say that the human chromosomal fusion, because it seems like the whole argument focuses on a fusion event that is specific to the human line. So it seems like it's providing, a very limited form of evidence. Because are you saying that it happened before or after the divergence from our common ancestor?
After our ancestors would have had. Our ancestors following the split with...
But at 48.
Ancestors would have had 48 chromosomes, and then the fusion occurred. But yeah, as Nesslig says...
Now,
but I do want to point out...
Okay, go ahead.
And this is in response to what we were saying, is that Nesslig is saying the chromosome actually has less nucleotides now than it has less now, not more nucleotides.
Are you saying there's not 150,000 more genetic letters? Are you saying there's not a huge difference?
He's saying it's smaller.
From my understanding, it's who's saying it's smaller?
Nesslig. He was one of the ones who helped me on actually putting together that video. So he knows quite a bit about the fusion.
Human chromosome 2 has 150,000 less genetic letters, you're saying, than the chimp?
We're just talking about the vestigial chromosome specifically.
Oh, okay, yeah. I think we were okay. So we're
not the entirety of it, just the vestigial part.
Is this telomeric DNA? Because normally it's found at the ends of DNA, right? But on chromosome 2, is this DNA like in the middle? Is it kind of mixed in the middle? This degenerate telomeric DNA?
Well, it's not in the middle. It's in the middle of one of the arms.
Okay, yeah, like because it's fused, right? So
so the two ends kind of rammed each other, and so you've got this little middle region on the arm that has the telomeric DNA.
So okay, so you're saying that this chromosomal fusion would have, because let's say our common ancestor had 48, the fusion happened, which gave us 46. So the human line is when it happened. So
right,
I don't understand how that's evidence for, because if it happened in the human line, why couldn't it, let's say after the flood, let's just say hypothetically, wouldn't that be enough time based on that bottleneck for us all to get the chromosomal fusion? And why not?
No, because again, you would have to go through orders of magnitude higher mutation rates, which even at AIG, creationists acknowledge that you're talking about creating huge waves of biodiversity.
Okay, so what if it happened before the flood? What if it happened before? So they already had it.
Then they would have
after the ark, and then that means that whoever came off the ark, they already had this chromosome 2 fusion. Then they passed it on to everyone else after that.
That assumes a whole lot. That assumes that we actually.
Well, you're assuming too, based on this.
Not actually, we can show that this is indeed what happened. This is the best. This is the most parsimonious explanation given the data that we have.
Well, the thing is, you're showing that this fusion, if it happened from a common ancestor, it happened in the human line. So it's not really demonstrating anything, significant about common ancestry.
Actually, what it's demonstrating
Yeah,
is
but
that
I just showed you an alternative explanation that could be true.
Because what your explanation would either have to say is that God created Adam and Eve with vestigial telomeres for God knows what reason, and I mean that literally, or they had 48 chromosomes, which then fused, and then that was passed along. Somehow it accrued, and somehow it magically accrued all of the mutations to make it degenerate today. Somehow.
Well, do we see chromosomal fusions today in human populations?
In humans, not really, but you can see fusion events in other animals.
Now, if there was
you can see the remnants of fusion events in other animals. Let me rephrase that.
But you don't see any type of fusion events going on.
Are not common things.
Now, if according to a model, if there is a degeneration process based on these, why would you assume that God created Adam and Eve with this, degenerated telomeric DNA? Why couldn't that happen thousands of years later before the flood based on degeneration? And then it get passed on to everybody after the flood bottleneck. Could that be possible?
I mean, with an omnipotent God, literally anything is possible.
No, I'm not saying he created them with that, with what we see now. I'm saying based on the degeneration process, because all I'm saying is the evidence from what I see is that it happened in the human line.
So that's
all we have.
Because
so now you're affirming the consequence. Because I think
So here's why it's not affirming the consequent. Affirming the consequent is a deductive fallacy. Science is not entirely deductive, if I remember correctly. It's science is an enterprise involving multiple. This, like I said earlier, this is not formal logic. Science is not formal logic. You can't say there's affirming the consequent in science because science is not formal logic. If you make a prediction and you say this evident, if we say, finding a fusion of two chromosomes in humans will validate my idea about our ancestry, and we do in fact find it, that's not affirming the consequent to say, hey, I found evidence supporting my conclusion. It may in fact be the case that there are other hypotheses that could be proposed which are better at explaining this data, even though it's predicted by this model. There are other potential hypotheses that could explain this, that could better explain this more parsimoniously.
But I'm trying to say, if they found there to be 46 chromosomes in, say, chimpanzees and in humans, that would be evidence for evolution. Now we see that chimpanzees have 48, we have 46. All of a sudden, that's evidence for evolution.
No, so it's a matter of parsimony.
Everything's evidence for evolution.
It's not, though, because technically it's a matter of parsimony. What you could say is that if there were a fusion event, if there were 48 chromosomes in gorillas and then 46 in chimps and humans, then what that would indicate to us is that this fusion event occurred before our common ancestor. And I mean, either way, you're still talking about what you're talking about is you're talking about inherited characteristics which are being passed onto offspring, and so we can still build these phylogenetic trees based off that. Because that's what passing characteristics. Because again, evolution is all about passing on characteristics.
And at the genetic level, we inherit the genes. That's why I believe that is where the debate is. But I believe that common ancestry is actually less compelling based on this alleged chromosome 2, because we actually see that difference, 48 versus 46 chromosomes.
But this was a prediction made based on common ancestry with other apes.
Okay. Did they see the 46 chromosomes and the 48 chromosomes first before the prediction?
Yes.
Was this, well, there you go.
Why is that? Explain to me why that negates the prediction.
That's where they affirm the consequent, because
how is that affirming the consequent?
Just because the grass is wet, it's not because it rains. So
remember what I said, how science is not formal logic. Affirming the consequent does not apply to science.
I'm saying regardless of the chromosome count in
No, that's not what
chimps
you're
versus
saying.
humans, it would have had to be evidence for evolution anyways, because that's the primary axiom.
No, what? Okay. First of all, affirming the consequent. If you were going to make a prediction and you say I found evidence to confirm this prediction, I can just turn that around and say, boom, you're affirming the consequent. But that's not how hypotheses work in science. There is no affirming the consequent in science
No,
because
I'm just saying it's affirming the consequent
it's
because
not.
it seems like you're saying it's the only conclusion, but I showed you
No,
that
I'm
there
not saying it's
is
the only
a
conclusion. I'm saying it's the most parsimonious conclusion.
okay, but you seem to, and I'm not saying God created. I'm just saying if there is the chromosomal fusion event, I think there's pretty good evidence of it based on the telomere DNA in the middle and based on the clear evidence of degeneration. But I wouldn't say that God would have created them with that, but based on the degeneration process, especially immediately post creation, if that chromosomal fusion event would have happened before the flood, more than
So
enough time for it to be
it's actually not.
inherited by everybody post flood.
No, you really need to look into what population geneticists actually say about uh, passing on, about mutation rates and the rate at which traits spread through populations, because a lot of what you said is misunderstanding that. But I do want to point out a comment Nestlig said. He said if Standing wants to argue that having two pair less chromosomes is evidence of non-common descent, then there are humans with 44 chromosomes. By the same logic, they aren't related to us.
Yeah, that's an interesting comment, Jackson. So, if you wanted to give some closing remarks here, brother. Honestly, I had fun with. I think this one was even longer than the first.
Probably.
I just at this point it's 2:30, and my wife and
No, yeah,
mate,
yeah.
I had fun. No animosity. I'd like we could probably do this a third time. We got some good information and good discussion in on this one, brother. Thanks for having me on the channel, eh?
Yeah, I enjoyed it. And I want to say thank you everybody for showing up. Thank you, Standing, for coming on. And it's also early where I am, or late, depending. But yeah, thank you everybody. And so I guess we're gonna sign off. Thanks for coming. One pair, not two pair. Okay, thanks. Alrighty. Well, thank you everybody for coming, and so we're gonna sign off. All right, bye.
Thanks, Jackson.